Hey, Daniel Kang here with Dr. Blair's BY4 through Immunology course. I'm going to be talking about clonal expression or selection and why it's important in the regulation of the immune system. So the immune system is highly regulated because it involves several different processes and instances where it needs to be very fine-tuned and very specific to certain problems like infection from pathogens and invaders. And those are taken care of by the use of white blood cells or leukocytes, or in this case, B cells and T cells. Most commonly, those are referred when people discuss an immune response. And as I've stated before, it's a very specific response to particular antigens and invaders, so that needs to be processed by the immune system to determine what needs to occur next. So even in the case of regulation, you don't want too much regulation or else the immune system can't function properly and it'll be more susceptible to invaders and pathogens. And on the other hand, you don't want it to be too little regulated or else just your immune system would be rampant and you have some sort of autoimmune disorder where you have a hyperactive immune response to even the most benign of pathogens. So what I'll be discussing is something, a phenomenon called clonal selection or expression, and this involves another two additional selections called positive and negative selection, depending on what occurs next after the initial selection process. So B cells and T cells have cell receptors that are specific for several different types of antigens, millions and millions of antigens that have been recognized or remembered from previous experiences or have been passed down from familial genes and stuff like that, acquired immunity, or passive immunity in that case if it's from a familial trait. But for the expression of a particular B cell or T cell, it has to be very specific. So if a specific pathogen enters the body, it needs to recognize that this pathogen is, first of all, destructive or dangerous, and it needs to proliferate to induce the production of more B cells and T cells for that specific antigen. So with clonal selection or expression, it involves the B cell and T cell, and these need to undergo recognition of antigens in the context of host major histocompatibility complexes molecules. These major histocompatibility complex molecules, or MHC molecules, contain the antigens that come from all types of pathogens, whether they be viruses, fungus, bacteria, parasites, things like that. And the B cell or T cell needs to recognize, or needs to be able to recognize the antigen on that. And if not, it's not going to form a proper immune response. It won't function the way it needs to, and it will fail to promote an action that can counteract the pathogen within the body. Therefore, if it can't recognize the MHC complex or cell molecule on that specific antigen from the pathogen, then basically the cell can't work. The B cell and T cell are faulty, they can't function properly, and they have to undergo apoptosis. That form of selection is called positive selection. The second type of selection that occurs during clonal selection for B cells and T cells is called negative selection. This is the case where once the B cells and T cells are able to recognize antigens and specific pathogens, they need to be able to control the expression and not overexpress in the case of a specific pathogen, whereas underexpression wouldn't be beneficial either. This is more important in the case of where there isn't an infection and, constantly the immune system is working to prevent, severe infection or anything of that nature to occur. But if it does happen without the presence of an infection, it would commonly be associated with some sort of autoimmune disease, and that's not good for the body. It's very damaging. So an example of this would be of antigens within the body that already persist, called self-antigens. They possess MHC complexes that are similar to the ones on pathogens, and that can be a problem because the B cells and T cells might not be able to differentiate between the two and cause the expression of and proliferation of more B cells and T cells to be formed when the antigen that's being recognized is not something that is of harm or of danger to the body. So the ultimate goal of negative selection is just so that the B cells and T cells can recognize self-antigens versus non-self antigens. And in this case, you want the B cells and T cells to recognize that non-self antigens are more of a problem, whereas self antigens are just within the body and can be recognized as non-problematic antigens. Now, the areas of where these cells proliferate or matured are different. T cells are commonly, they all originate from the bone marrow, and T cells, immature T cells, will travel to the thymus, the lymphoid organ, and will proliferate and mature there once they pass both negative and positive selection. And B cells will actually proliferate and mature in the bone marrow. So that will be expressed and be matured in a different area compared to the T cells. So finally, through this process, we can determine how B cells and T cells are able to finally tune into specific antigens and recognize that these are such foreign invaders and they need to be taken care of in an immune response manner. And in the case of autoimmune diseases such as cancer, HIV, or AIDS, this is a problem where clonal expression/selection hasn't been as efficient as with a normal healthy human being, and it can cause problems within the body and cause an overexpression of the immune system, which causes damage and in some cases death. So got to be careful. Thanks for listening to me, and I hope you learned something about clonal selection.